Happy morning. I feel extremely glad to welcome you all for this revision section. Let's explore knowledge. Lesson 6, Cell, the unit of life. It has got 5 answers and 5 choose the correct answer. So we'll start with our 5 answers now. Question number 6. Bring out the significance of phase contrast microscope. So you're going to write the importance of the phase contrast microscope. The answer is phase contrast microscope is used to observe living cells, tissues and the cells cultured in vitro during mitosis. Okay, so what happens during the mitosis? Those details of a cell can be observed using the phase contrast microscope. So phase contrast microscope is used to observe the living cells and the cells cultured in vitro during mitosis. The second question, 7th question, explain the protoplast theory. State the protoplast theory. Okay, this protoplasm theory was proposed by Maxwell, which states that the protoplasm is the living content of a cell and it is a complex colloidal system. Okay, it has got 2 points. Okay, first is living content of the cell and the second thing is it is a complex colloidal system. Okay, so protoplasm theory proposed by Maxwell and it states about living content of the cell and is a complex colloidal system. So this makes your protoplasm theory. Then 8th question, distinguish between prokaryotes and eukaryotes. Okay, we are going to make a differentiation of both the prokaryotes and eukaryotes. You can find them taken in a table with 3 columns. One is about the features, the second is prokaryote, the third one is eukaryotes. Okay, so it has got some 9 features. Okay, with the 10th point as the example. Let's look into the features one by one. Size of the cell, nuclear character, DNA, RNA protein synthesis, ribosomes, organelles, cell movement, organization, cell division, example. So these are the 10 points that we have taken for the features. Now let's move to the characters of prokaryotes and the eukaryotes. First thing about prokaryote. The size of the cell is approximately 1.5 nanometer and eukaryote it's approximately 10 to 100 nanometers. Okay, this is about the size. Nuclear character. Prokaryotes have nucleoids. They don't have true nucleus. True nucleus are absent. They have only nucleoid. Eukaryotes, they have true nucleus with a nuclear membrane. So this is the difference between prokaryote and the eukaryote. The third difference, DNA, usually circular. Okay, the prokaryotic DNA is usually circular without protein. Histone protein is absent. Whereas in eukaryote, they have a linear DNA structure with histone protein. Is that clear, children? So DNA, it is circular. In eukaryotic, it is linear. And here it is without histone protein. Prokaryote has histone protein. So that is the difference between prokaryotes and the eukaryotes. Now moving to the third point. RNA or protein synthesis. Okay, it takes place by couples of cytoplasm. Cytoplasms come together and in the cytoplasm it takes place. Whereas in eukaryote, RNA synthesis is inside the nucleus. Okay, it is inside the nucleus and protein synthesis is in the cytoplasm. So RNA is in the nucleus, protein synthesis in the cytoplasm, whereas both your RNA synthesis and protein takes place in the cytoplasm. Fifth point, Ribosome. The ribosome of prokaryote is made up of 50 S plus 30 S. The larger unit, subunit are there, no? Larger subunit and smaller subunit, it is 50 S plus 30 S. Whereas in a eukaryote, it is 60 S plus 40 S. It is 60 S plus 40 S. So that is the difference. Organelles. In prokaryotic, organelles are absent. In eukaryote, you have found many numerous organelles are present. Cell movement. The prokaryotes moves with the flagella. Eukaryotes moves both with flagella and cilia. Organizations usually single cell. Okay, they are made up of a single cell. That is why it is prokaryote. Eukaryote, it may be single or colonial or multicellular. It may be of three forms, that is single, colonial or multicellular. Cell division, usually binary fission, whereas here it is mitosis and meiosis. Examples, bacteria and archaea. Here the example is fungi, plants and animals. Children, you got 10 points. If you get this answer, if it is in a 5-mark detail answer, try to write all the 10 points. Isn't clear? So, all write all the 10 points. So, this is about distinguishing features of prokaryotes and the eukaryotes. Now, let's move to the next question. Question number 9. Okay. Difference between plant cell and animal cell. Right from the lower sections, you will be learning about it. Plant cell and animal cell. Plant cell, usually they are larger than animal cell. Animal cells usually smaller than the plant cells. Cell wall present in addition to plasma membrane and consist of middle lamellae, primary wall and secondary wall. So in plant cell you got a cell wall, whereas in animal cell wall is absent. Apart from cell wall, they also have a membrane called the plasma membrane. This plasma membrane consists of middle lamellae, primary wall and the secondary wall. Third point, plasmodesmata is present. Plasmodesmata is absent. It is a connecting tissue. Chloroplast is present, very important. That is why plants look green in color. Animals, chloroplast is absent. And next point is vacuoles, large. Here vacuoles very small. Vacuoles are permanent. Vacuoles are temporary. So vacuoles are large and permanent. Vacuoles are small and temporary. The last point would be tonoplast present. So these are a lysome-like structure which is present around the surrounding, a layer around the vacuoles. Here tonoplast is absent. Lysosomes are very rare. Lysosomes are present. So altogether you have got some 7 points for the difference between plant cell and animal cell. It is large, it is small. Cell wall present, cell wall absent. Plasmodesmata present, plasmodesmata absent. Chloroplast present, chloroplast absent. Vacuoles are large, vacuoles are small. Tonoplast present, tonoplast absent. Lysosomes are rare, that is they are not that much present. Lysosomes are present. So these are the difference between plant cell and animal cell. Now let's move to the, you got some more difference too. Centrioles. Centrioles are absent except motile cells of lower plants. Here centrioles are present. And the storage material here is a starch grain, and the storage material in animal cell is a glycogen. And nucleus are present along the periphery, nucleus are present towards the center. Children, if it is asked in 3 mark, write only 4 to 5 points. If it is asked in 5 mark question, try to write all the points. Now let's move to the last question, very interesting and a very important question also. Draw the ultrastructure of plant cell. Draw the ultrastructure of the plant cell. So this is a plant cell. You will have to draw it and label the parts. We'll start with the first part, that is with the cell wall. The outermost lesion is called the cell wall. Followed by the cell wall, you will have the cell membrane. This region is called the cell membrane. Next you got the nucleus. The nucleus is present with the peroxisome inside. And this is mitochondria, vacuole, cytoplasm, ribosomes, Golgi apparatus, chloroplast. So these are the various parts that you find in the structure of a plant cell. This is a very important question, children. Try to draw and practice this answer. I hope you would have enjoyed learning the answers of lesson 6. Revise those answers and get prepared for your exam.